I mean, that's the thing, right? So you think about plastics alone, right? Like,
oh yeah,
you know, all the things, yeah, right. Lots of focus on the negativity, but like we can, you know, you can go and have surgery now and not get it. Is it time to start?
That's fine. I believe it is.
Okay.
Okay. And the excuses, we're not finished. Thank you. Okay.
Do you want to use my mic? Just in case you need.
Oh, I don't think I need the mic, but okay. Good evening and welcome to another edition of the Evening at Skidaway lecture series. I want to thank all of you in person who braved the elements to come out and be with us here tonight. And if we have folks online viewing us on our YouTube stream, welcome as well, and thank you for joining us. Tonight, we're going to be hearing from Dr. Daniel Ohnemus, who has a Bachelor of Arts in Biology and Chemistry from Williams College from 2004, and a PhD from the Massachusetts Institute of Technology Woods Hole Oceanographic Joint Program, where he got his PhD in 2013. And after doing postdoctoral studies at Woods Hole Oceanographic Institution and at the Bigelow Laboratory for Ocean Sciences, he joined us here at Skidaway in 2018 as an assistant professor. And he will be telling us about his research program that he's been building since that time. The last thing I will say, I see that a lot of the folks in the room are already members of the Associates of Skidaway. But if you would like to get on our mailing list and are not on it already, we have a sign-up sheet here at the front of the room. Please give us your name and email address, and you'll get our email newsletter, which comes out quarterly. And if you're interested in joining our Friends organization, there's a envelope where you can join our group, which supports Skidaway Institute's activities, such as this evening lecture series, in the copies of the newsletter that are up here on the table as well. So with that, I give you Dr. Dan Ohnemus.
Thanks, Lark. Thanks, everyone. Get my lapel back on. Yeah, okay. I can't see or hear the YouTube stream, so I assume it's amazing. Okay, so tonight I'll be talking about geotraces. The subtitle here is How Mapping Ocean Chemistry Helps Us Learn About a Changing Planet. But in general, as Clark mentioned, Geotraces is a program that I've been involved in basically since I started as an oceanographer in the joint program at MIT. My first year in the program, my PhD advisor was basically funded to start doing one of the first sampling programs as part of the Geotraces program. And it sort of followed me through my career. Mariah, my graduate student, and I sailed on an expedition just last winter as part of Geotraces as well. So it's made up a big part of my career, and I think this is a great opportunity to talk about all the different and cool things that I think Geotraces does. So Geotraces is a chemistry-focused program. It's basically designed, implemented, and run by chemists. And so I have some questions for a public audience, namely, how many of you disliked or maybe liked chemistry when you were in high school or in college? Show of hands, were folks generally like favorable on chemistry if they raise their hands? Okay, good, my boss is raising his hand, so that's good. And then folks that maybe disliked chemistry? Yeah, okay, yeah, some of my colleagues and peers.
Like me, what's chemistry?
Right, yeah, and people who just have never even taken it or don't even care to understand it, right? Okay, so that's interesting, right? Like chemistry often has this place as a mixed opinion. Even my undergraduates that are taking my own classes often say that chemistry is the part of their oceanographic experience that they're most loath to embrace when they start taking all these interdisciplinary courses. And so I myself, as Clark mentioned, I was also a split understanding here. I did biology and chemistry. I couldn't pick one of them as an undergraduate. In the long run, I drifted more towards chemistry, but I can tell you that my grades in college were much better in biology, in part because chemistry was very rigorous. Yeah, so related question, we're gonna be talking about the chemistry of the oceans. What sort of comes to mind when you think about ocean chemistry? Is there any like terms or things that pop up in your brain for it? I'm sort of curious whether they're gonna jive with what I'm talking about in the talk, so. Any thoughts?
Acidification.
Acidification, okay, yeah, so like the pH of the ocean, right? That's a big part of the chemistry, for sure.
Salinity, dissolved minerals.
Yep, salinity, minerals, things that are dissolved in seawater. Yep, those are all really critical aspects of ocean chemistry. Actually, those two things alone encompass huge areas of research. And actually, most of the things I'll be talking about today are things that are minerals or formerly minerals that are dissolved in the oceans. So yeah, so I think that's sort of on track there. I see a hesitation here.
Chemistry is what makes the ocean live?
Yeah, so its role with biology, for sure. And actually that is, in many ways, the driving part of Geotraces, as we'll see, is that the chemistry is intricately linked to the biology of the oceans, which organisms can grow where and how much they can grow.
Well, that's easy for you to say.
Good to know. Okay, so yeah, so those sort of terms are all gonna be coming up here. So I'll give a quick definition. So Geotraces is broadly a program focused on mapping and understanding the chemistry of the oceans, and specifically the low abundance or trace elements that are in the oceans. So in order to talk about those things, we'll need to have a little bit of a crash course in ocean chemistry, which I hope you'll bear with me for. And I'll try to keep it light, no equations, no stoichiometry or algebra, or the things that maybe led you to getting a B- in kinetics like I did in my senior year of college, right? Okay, so none of these things are gonna crop up here. Broad overview of this program. It's been going on since 2007. And as I mentioned, I started graduate school 2008. So since 2007, over 900 scientists, graduate students, senior professors, technicians, helpers all across the world, scientists of all types have been involved in collecting all these samples, analyzing and generating data about them, and interpreting it. This includes, it's an international program, so over 36 nations have either funded or led expeditions as part of Geotraces. This includes landlocked ones. We have colleagues in places like Poland, countries that are interested in ocean chemistry that have no direct connection to it. I think that's interesting. And also there's over 120 expeditions which have sailed in order to collect these samples. And also Geotraces has inspired, because it's been going on for over a decade now, almost two, many other programs that have sailed as a result of it. So, and some of them are starting to use Geotraces compliant sampling methods. So the things that we invented in order to make these measurements are now part of sort of the ocean vocabulary just for taking samples. So it's had a wide influence in a short period of time. Most of Geotraces data is collected in these lines across the oceans here called sections. The ones in black and yellow are sections that have been completed by this international effort, so spanning the globe between all the major continents in the poles, in equatorial regions. And then there's still, all the sections in red are ones that have not yet been completed. So these are efforts that are, most of them are currently pretty well on in their planning stages, but some of them are quite large, right? Big sections of the South Atlantic that aren't analyzed, sections in the Indian Ocean, and wide swaths of the Pacific, which have yet to be analyzed. Sure, question?
How how do those sections get decided?
Oh, that's a great question. So in part, it's based on where earlier sampling efforts have been. You'll notice that some of the lines pass through major ports of call, so places where it's relatively inexpensive to ship gear and to lead an expedition out of. And so there's a lot of hysteresis, like the historical ocean sampling. This line specifically, along 150 degrees west, has been sampled since the 60s, fairly regularly. And so Geotraces tried to overlap and expand in areas that we thought might need that type of coverage. So yeah, it's a great question. So we call these expeditions cruises. I sometimes might call them that. I just want to reiterate that it's probably a misnomer. They're not very cruise-like. There's lots of sampling, very little sleep, very few ports of call, if any. I don't think I've ever been on one with a good port of call. The other thing that's sort of interesting here is that because this is a chemical program, unlike some of the biologists in the room, we actually generally don't care what time of day it is. So if we get to our sampling site and it's 3 in the morning, the chemists are going to put and start sampling right away. They're not going to wait till dawn, till all the organisms wake up in order to get sampling there. So this is very different from some of the past biological programs where time of day is an important thing. My grad student can attest to wake-up calls at all hours of the day. So yeah, so these sections have brought us to lots of different places. These are some of the US Geotraces sections, maybe the more extreme ones. One that went to the North Pole here on the far left, the Arctic section, GNO1, which involved sampling on the Coast Guard cutter Healy there. Huge Coast Guard complement plus scientists, almost 100 people on board there. And then some of the sections in more remote places like the Central Pacific, this section here, GP15 had a smaller complement, no Coast Guard, but about 30 scientists on board collecting samples. This picture will come back in a moment here, so don't forget about it. But yeah, so this is my PhD advisor there. I'm not in any of these pictures. I applied for funding for this cruise and got rejected, so I didn't go.
Can I go back?
Sure.
Along the United States and the Gulf
Mm-hmm.
and South America,
Oh yeah, this one here.
all in red.
Yeah.
Is that because it's so complicated?
That's one section. So this little section in the Gulf of Mexico, the one that's like in the, it's hard for me to point.
Use the stick.
Yeah, here we go, it's a stick. Yeah, so this section here.
Yeah.
Yeah, so that's really new. They only added that like last year in response to a growing understanding that the Gulf of Mexico is really important in setting North Atlantic chemistry. So yeah, this section is like the least developed of all of them, in part because it's so complex and so near shore. So yeah,
Wow.
yeah, really interesting.
So close.
Yeah, it's so right. You think it's right nearby, right? Yeah.
Is that because that the people don't flush their toilets?
I guess we'll find out once we sample it.
No, that was-
Okay, so Geotraces not only is these long sections, but it's also involved all these different technical challenges to making these measurements. So one of the technical things that will come up here is that most of the chemicals we're trying to measure are very, very low in concentration. For instance, so iron is a key parameter of Geotraces. The typical iron measurement in the open ocean is the amount of a single paperclip, in this case a Prada paperclip, which is worth $185 I found online. A single, Prada makes paperclips now, yeah. So one single paperclip dissolved in 30 ocean Olympic-sized swimming pools, right? So you take that paperclip, dissolve it in 30 swimming pools, take a liter sample of that, and then measure how much iron is in it, right? It's a very technically challenging problem. So that's one example there. Thorium and radium are two radioisotopes that are measured. Some of the measurements for these elements include measuring just a few atoms of these elements in a sample that's over 1,000 gallons, right? So you have a 1,000-gallon sample and you're measuring only a couple atoms decaying every few minutes in that sample, right? Very technically challenging processes here. And then all along, right, we're doing this in oceanography, we're doing it on a big rusty ship. So this is the corner of that picture before, right? This is the Ravel. Ship that I just spent a significant amount of time on myself. And you can see that the ship itself is actively trying to contaminate almost everything we do, right? These rust spots, even a well-maintained ship like the US fleet generally is constantly shedding iron particles into the ocean. So really complicated problem. So one of the first things we do when we go to sea is build this thing called the bubble. It's a little YouTube video about it. Actually, I'll just leave the audio on. I don't know whether it's going to get picked up. But in general, we basically go to sea and we cover everything that we're going to be doing our sample collection with in plastic here. So we hang these sort of string here. We take these giant sheets of plastic that we then stretch across the ceiling and hang over those sort of string there. This is everyone's gear, which is not yet put away because no one can put it away until the room is clean, right? Then we use an exorbitant amount of packing tape to splice all these sheets of plastic together, right? This is a big effort too, right? So this is like 5 or 6 people working for several days to do it. And actually do this process in port before we go to sea, which is nice. And then we have Seth John, collaborative of mine, and I presume this is his graduate student based on what you'll see he does with his back here. So then we hang these high efficiency particle filters, these HEPA filters, up on the ceiling, and then we cut them into the plastic. And this basically acts as a high particle-free air supply into the now plastic bubble, where we'll collect and analyze, in some cases, all of our samples. So after those HEPA filters get turned on and the packing tape has done its job, the bubble starts to inflate. So that's why we call it a bubble, right? So all this basically particle-free air is now filling the space. The bench top is covered in plastic. Yeah, and we do leak checks and stuff like that. And then at this point, most of the space is relatively clean or cleaner than it was before in order to collect clean samples. So yeah, positive pressure. So it's like having we basically build a big clean room at sea. And Geotraces is crazy. Oh yeah, question here.
Do you run control samples in that environment before you?
Oh, definitely. Yeah, so we have particle samplers. We just so the HEPA filters are really rigorous. Like they're one of the like sort of miracles, I guess, of oceanography is that it's really hard to mess up one of those HEPA blowers. And so if you're working directly under or around it, you're pretty clear that the air itself is not going to be contamination. At this point, most of the contamination actually is likely to come from you, right? So from a dirty glove or from sweat leaking out of your hand or something like that. So yeah, Geotraces is a big part of it. I would say at least a third of the samples collected are usually blanks in order to check that background over time. Yeah, so.
Ships
Mm-hmm.
are already equipped that a ship is not already equipped.
Yeah, so that's the thing, right? These vessels are, Geotraces was in sense invented after these vessels were laid down. And so clean spaces are not a normal part of oceanographic vessels. It's something that we've had to invent in order to do the science.
Because spaceships, everybody wants a nice spaceship.
Yeah.
Nobody gives a damn about the vessel.
Right, yeah.
There's no money.
Right, they want a vessel that floats, right? So there are mobile clean vans. So if you're doing a smaller sampling operation, say a 1 lab, and you're going to sea and want to have a clean space, you can request basically a small trailer that has a clean space pre-built into it. And some of the Geotraces sampling happens in a van that's specially designed and is basically arrives ready to go. But Geotraces, and this is what this slide says here, on Geotraces we're measuring all these different elements in blue, right? So some groups are measuring like all these rare earths. Others, like my group, are measuring more of these transition metals up at the top of the periodic table. And so the different groups have different requirements for what is clean for them, right? It's really easy on a ship to contaminate for iron, but much harder to contaminate for terbium, right? So different groups have different levels of cleanliness, and we have to work together to make sure that everyone's needs are met. It's a big, complicated problem. So generally, we just try to be as clean as absolutely possible and hope for the best, right? The blanks will tell you if you did it wrong. Okay, so why are we doing this, right? I said we're going to talk about chemistry. So I want to talk a little bit about the chemicals themselves and what they do, right? And they sort of fall into 3 major roles here, right? Some of these elements that I showed on the previous slide here act as nutrients, right? These are elements that support the growth of phytoplankton, the base of the food chain in the oceans, and also support higher organisms and the ecosystems themselves, right? So many of these elements act as nutrients. We can sort of split the nutrients. Oh yeah, I use this analogy of a land plant a little bit, but in the oceans, we'll be thinking more about these sort of smaller microscopic organisms, diatoms, okay? So nutrients have sort of 2 major styles. There's macronutrients, the things that are available in much higher abundance, things like nitrogen, phosphorus, or silicate. If you have a garden and you fertilized your garden before, you might have put fertilizer down that has nitrogen, phosphorus, and potassium in that case in a soil environment. These are things that plants need a lot of in order to grow. But also every organism needs micronutrients, things that they need in small amounts, but they do need, right? No organism on Earth can live without a little bit of copper and a little bit of zinc in it, right? And different organisms have different requirements for how much they need, right? An organism like a whale needs a lot more zinc and copper than a small phytoplankton, right? So some of these things scale with the size of the organism, and some of them don't. Some organisms have really high iron requirements because they do chemistry that requires a lot of iron. Other organisms might need basically no iron at all. They've streamlined their whole ecology around having small amounts of iron. So ecosystems are really complicated, and basically the more rare an element gets, the more variable its needs might be in the ocean. Okay, so these are the nutrients. There's also other things that Geotraces measure, so contaminants. So these are elements that might be derived from humans or anthropogenic. And these types of contaminants, they might inhibit the growth of organisms or poison them. They can alter the chemistry of an ecosystem, right? Select for one organism over another, say an organism that has the ability to deal with a toxin and one that can't. Also, some contaminants from human activity can even enhance ocean productivity. So things like biomass burning, burning a forest and generating a lot of smoke. Some of that smoke might contain elements that organisms actually need. And so contaminants can sort of also act as nutrients in some cases. Another sort of thing to mention here is that sometimes things that are micronutrients, like cadmium, can be toxic at higher concentrations, right? Like you can have a glass of milk with breakfast, but if you drank 2 gallons of milk, it's gonna make you sick, right? So some things in small doses, the dose makes the poison, right? A lot of what Geotraces is trying to do is understand for all these different elements in the periodic table, which ones are contaminants, which ones are nutrients, and where are they acting like that? It's a big open question in the way the oceans work. Okay, and some of the things we measure are tracers. So these are elements or isotopes of elements that give insights into complicated processes. So things like ocean circulation, or the exchange between the atmosphere and the ocean, or the ocean and the sediments at the bottom of it. A lot of those interactions are really hard to see, right? We can't actually, with a satellite, look at how the ocean is interacting with the dirt at the bottom of it. But if there's an element or an isotope that is a signature of that process, and we can collect that element, then we can see where and how, to what extent the ocean and the sediments are interacting, right? So a lot of what Geotraces is doing is learning about, for all these different elements, which role they're taking, and also trying to discover new ones and measuring the ones we knew about. Okay, so let's talk a little bit about some, so I'm going to provide a little example of each of them before I bore you all to death with chemistry, right? So let's talk about these nutrients, these things that support growth, right? In order to understand this, we need to think about this thing called Liebig's barrel metaphor, or Liebig's law of the minimum here. So has anyone heard of this before? Is this totally new to folks? Okay, totally new, right? So what Liebig, and Liebig actually didn't invent this, he popularized it. Someone else invented it, classic science. So what Liebig decided and popularized in regards to agriculture and soil, this is not about the ocean, but it does apply to it, is that the yield of an ecosystem, how much biomass can grow in an environment, in this case, in his case, soil, or in our case for this talk, the oceans, is limited by the availability of nutrients. And it's not actually the total amount of nutrients that are in the ecosystem, it's the most limiting nutrient, right? So if all these different staves of the barrel are different things that a plant needs to grow, things like light, water, soil, or all these different elements of the periodic table, it doesn't matter if you have 2 tons of nitrogen and perfect amounts of light and just the right amount of water and humidity. If your soil has no iron in it, nothing's going to grow, right? So the lowest stave in Liebig's barrel is the one that decides what the ecosystem can do, how much that ecosystem can do. A little shade of meaning here is also that different ecosystems sometimes evolve when they're limited by different things. So a phosphorus-limited ecosystem can look very different from a nitrogen-limited ecosystem as far as which organisms can grow there. And micronutrients are the same way. So we're still trying to understand where and how much of these nutrients are available, and therefore what can grow in the ocean and to what extent. Yeah, so I made this point before. Nitrogen is often the most limiting nutrient. For most of the world oceans, it's the most limiting nutrient, nitrogen. So before we talk about these sort of other rare elements in the periodic table, I wanted to talk about and look at some of these macronutrients, the things that you might be familiar with from soil, right? So a couple of things to look at here. So this is a nitrogen profile in the oceans. We're looking at a profile from the Atlantic and the Pacific. So more nitrogen is over here on the right, and the surface of the ocean is up here, and the deep ocean a couple miles down is down here, right? So let's just look at this Atlantic profile here. So what do we see? So the nitrogen is actually very low at the surface, right? Both of these profiles, both in the Atlantic and the Pacific, have very low amounts of nitrogen up at the surface where organisms are growing. Why is that the case? Any ideas? Why might these nutrients be low at the surface? Yeah, you think it would be there around, right? Yeah, but what's happening, right? So organisms are growing, right? And what are they doing as they're growing, right? They're consuming these nutrients from the environment, right? So at the surface of the ocean, where all these phytoplankton are growing, things like nitrogen and actually also other nutrients like phosphorus and silicate, which acts as a nutrient in the ocean, they all have the same shape, which is that they're being consumed by organisms at the surface and regenerated down at depth. So about 1 kilometer down, almost all these nutrients come back to much higher concentrations, right? So this is interesting. There's something going on here as far as macronutrient chemistry, which is common across all these macronutrients, okay? So another thing that's interesting that I'll point out here is that most of these macronutrients, nitrogen, phosphorus, and silicate, are very long-lived, right? So they're very stable in seawater. They're happily dissolved. They can act as nutrients for many thousands of years. They can get used by one organism, recycled, brought around the ocean, and come back for another try, right? So most of these elements have residence times on the order of tens of thousands, if not hundreds of thousands of years. So they're being basically consumed and recycled many times. The other thing is that most of these macronutrients, these most important nutrients, were measured in the 70s and through the 90s in these programs called GeoSex and WOS. GeoSex was actually named in order to sound dirty. So that was part of the thing in the 70s. Interesting times. Okay. But in general, these macronutrients, these things that Liebig identified as soil as being really important very early on, were mapped pretty early in oceanography. But these lower abundance things, the micronutrients, were not as well mapped early on. Okay, so just to summarize what we saw on that previous slide in more of a cartoon form, right? So we have photosynthesis producing biomass and oxygen at the surface of the ocean, right? I lied, there's an equation here. So it takes nutrients and CO2 and water in the presence of sunlight, and you make biomass, right? So this is all these happy diatoms that are growing in the presence of these nutrients, right? But then what happens when they die? They sink out, right? So what happens if you don't fertilize your garden, or if you don't water it, or if you do something like grow a mustard plant in Savannah, which I tried to do last year, right? They are not happy around here. Okay, what happens when you don't feed a plant on land? It wilts and dies, right? But like, what physically happens to it, right? Your big happy like apple tree that you mistakenly poison or something like that, right? It actually like falls to the ground, right? The plant wilts and decays, right? It can no longer support itself. And phytoplankton are no different. So organisms that get limited by nutrients or that just sort of go through their natural process of being born and being eaten, for instance, also sink out. But in the oceans, they don't just fall into the soil and get recycled. Their bodies sink into the deep ocean. So that's why these profiles on the previous slide here have a maximum down below, is that regeneration that brings those nutrients back into seawater doesn't happen at the surface. It happens down in the deep, dark part of the ocean. And on average, it happens like the maximum for it reaches at about 1,000 meters down. So this is an interesting thing here where photosynthesis is consuming nutrients, respiration is reproducing it, but down below. And also, this process is really hard to measure, and I'll make a pitch for this right now because we'll talk about it. We'll see how Geotraces has addressed this later on. This process, called the biological pump, is very difficult to measure, in part because it's very slow, and also it's very variable, right? Measuring it right here and measuring it 10 miles away, you might get a totally different number on any given day. Okay, so what's connecting all this stuff together is the physics. So chemistry is operating between the biology, the organisms, and the physics that are moving all these nutrients around. And you may have seen a figure like this before. So part of the reason that some of these macronutrients were higher, and all of them were higher in the Pacific than the Atlantic, is that over time, the respiration is allowing those things to build up in the deep ocean. Because the deep ocean is basically formed only in these regions where deep waters are formed. So where cold, dense waters sink and fill the deep ocean. So this sort of thermohaline circulation, which is moving the water mass around on the order of about 1,000 years, allows these macronutrients, the long-lived nutrients, to increase in the deep ocean over time. And we see them higher in the Pacific, where it takes about 1,000 years for that water to reach. So there's this whole sort of interesting chemistry with macronutrients, which was understood before Geotraces started. Okay, I just made these points, about 1,000 years. Okay, so what about these micronutrients, all these other things that Geotraces is trying to measure? Wouldn't it be great if they were all like macronutrients, right? If they just behaved like macronutrients. They hung around for a long period of time. They were higher in the Pacific than the Atlantic. They were low at the surface and increased down below, right? That would make all of our chemists' jobs very easy. But it turns out they're not, okay? So this is a classic micronutrient here. And if you went to Clifton Buck's talk last month, you probably heard a lot about iron. Okay, so these are two profiles of iron in red from the Atlantic and in blue from the Pacific, right? The shape is totally different here, right? In one of these basins in the Pacific, the concentration is pretty low at the surface like a nutrient, but in the Atlantic, it's pretty high. It's actually elevated, right? None of them have this big bulge at 1,000 meters, right? Only the Atlantic profile has this sort of maximum down here, 3 kilometers down, right? So they're behaving really differently. The other thing that's sort of cool here is that the basins sort of match, right? Most of the profile, with the exception of the surface and down below, is about the same, right? So for iron, this important nutrient is not accumulating from the Atlantic to Pacific like the macronutrients are. So something's really different about its behavior. Any guesses? Why does iron behave differently in seawater than something like nitrogen or phosphorus? You actually already have an intuitive. It's not its magnetism. What would happen if you put an iron bar into the water nearby here or hung it off the dock? It would rust, right? Yeah, remember that rusty ship, right? The reason they're always painting the Ravel, except they don't paint it during Geotraces. Most of the time when you go to sea, they're constantly painting the ship and it's super annoying. But yeah, iron rust, right? So it forms particles. So it behaves really differently from these other nutrients. So this is a microscopic view of what happens to iron when you try to dissolve it in seawater. Oh, it forms these little micro nanoparticles that basically they're still in the seawater, but it's almost like a micro rock, right? So plants and organisms can't access iron when it's locked in this sort of particle forms. And so it can act as a nutrient, but only when it's available and only for a very short period of time. So in general, a lot of these micronutrients that Geotraces is trying to measure have very different chemistries. Some of them behave like this, where they precipitate and dissolve and are inaccessible. Other ones act more like macronutrients and are somewhere in between. So there's lots of different chemistries going on here, which is why we have to sort of study them all individually to make sense of what's going on. Okay, so that's a nutrient. We also, Geotraces, like I said, has done all these different sections, right? So this, you'll see, if you Google Geotraces after this, which maybe you won't, maybe you'll be totally bored by it, but this figure comes up like all the time. Everyone is so proud of this figure. They spent decades planning this program in order to generate figures like this, right? So we went from having measurements of iron only at Bermuda and only from the early 1970s to having all of a sudden data about iron for the entire Atlantic Ocean, right? Thousands of data points collected, hundreds of people's lives and effort going to collect these data. And all these different things start to emerge. Same things that we saw when we looked at the individual profiles, right? In some cases, the iron is low at the surface and acting like a nutrient. In other places, like off the coast of Africa here, iron is really high. It's crazy high, right? It's way higher in abundance than organisms could possibly use. And then also we have these big tongues, right? So remember that big maximum at 2,000 meters? So these are hydrothermal sources. So basically the crust in the deep ocean is opening up and venting some of these nutrients into the deep ocean directly. So there's all these different things happening in the micronutrients that are not happening in these other better studied nutrients. And all of a sudden there's all these different roles. So things like all this iron signal from Africa is coming from dust that's blowing off the African continent, right? In the South Atlantic and also in the Southern Ocean, it's obscured by this little map here. These hydrothermal sources of iron are super important. And some of these data, we measure iron when we're actually at sea. So we come to the end of these cruises knowing something about iron that no one has known before because we've never measured in that location. One of the things that has emerged about iron is that it's still not clear whether these huge bullets, these tongues of hydrothermal iron, or to the extent of this dust which is being deposited, how much of this is actually influencing biological growth, right? Because we saw before that iron likes to sink out and precipitate. So how much of it can organisms access? We really don't know. We're only mapping it at this point. A lot of interesting questions that are emerging from just having these data in hand. Okay, so these are nutrients, right? These are things that organisms like and really want to have access to. So what about the contaminants, right? I mentioned some contaminant elements. So let's look at lead. Lead is a metabolic poison. Most organisms don't want any lead at all in them at all. It doesn't act as a micronutrient, at least to our knowledge. And we see these are some profiles, again, from the Atlantic and the Pacific in blue. And there's totally different things going on here, right? And this is a section here. So this is basically looking at, in color coding, the Pacific Ocean here with low concentrations and the Atlantic Ocean over here with the warmer colors. So any ideas why lead, this contaminant element, is higher in the Atlantic and basically completely absent in the deep Pacific? What do you think humans, what did humans do?
It's coming from land sources originally.
Yeah, so yeah, it has a mineralogical
There's a
source.
difference in industry over time
Sure.
in those two oceans.
Yeah. So it's a human contaminant for sure. Anyone? I think there's enough gray hair in this audience to remember. I remember this source as well. You probably fueled up your vehicle with it.
Yeah, leaded gas.
Yeah, leaded gasoline, right? So the reason this contaminant element is higher throughout the Atlantic and low in the Pacific is the use of leaded gasoline. So we started adding tetraethyl lead to gasoline in the early 1900s. It's an anti-knocking agent. It makes engines behave more cleanly, burns the fuel more efficiently. But also it's a toxin, right? We found this out in part due to increased crime during the 60s, 70s, and 80s, right? Leaded gasoline is associated with negative outcomes in human behavior as well. It also acts as a toxin in the oceans. So the reason it's higher in the Atlantic is that this use of leaded gasoline, and by the US and European countries which surround the Atlantic, reached a peak in the 70s before it was basically banned in the early 80s, okay? So leaded gasoline, and one of the sort of founders of Geotraces is this scientist, Ed Boyle from MIT, who made a career studying lead. And he'd been making measurements at Bermuda for quite a while and saw these lead concentrations beginning in the 1970s basically decrease and enter the deep ocean through this sort of particle sinking action in part over the course of time until the, I guess this latest data point, the purple curve, is from 2011. So banning leaded gasoline basically stopped having it be added in the surface of the ocean, but the signal that was already there basically gets moved by circulation and biological processes into the deep ocean. And because that leaded gasoline and those fumes and the aerosol deposition mostly happen in the Atlantic, the concentrations in the Pacific are significantly lower. So that ocean has a much lower signal of it. The role is reversed for other things, right? So some of the radioactive elements that were set off in the bomb testing during the 50s and 60s have maxima in the Pacific because that's where many of the bombs were set off and where most of those aerosols were deposited. So human effects can be much more spatially specific than some of the nutrient effects, depending on what the element is. So pretty cool stuff to be learned from measuring something like lead. Okay, last final example before we look at some cool Geotraces pictures. So I mentioned before the biological pump is really hard to measure, and some of these elements, these tracer elements, can be used to answer tough questions. So the example I'm going to show here is this basically parent and daughter pair. So uranium-238 is an element that has a half-life of 4.5 billion years. Okay, and it decays to a daughter, thorium-234, which has a half-life of only 24 days. Okay, so super long-lived parent isotope and a very short-lived daughter isotope. Okay, oh, where'd my little explanation go? Okay, so another little thing to mention here is that uranium is very soluble. So, and I'm not talking about contaminant uranium from nuclear reactors. I'm talking about just natural uranium that's present in the ocean. So we're not talking about contaminants anymore. This is just natural uranium in the salt of the oceans. So uranium is very soluble. It's dissolved in seawater almost everywhere in correlation with salt. And it's entirely naturally sourced. So it's constantly decaying to this daughter, thorium-234. And so what we might expect is that in the oceans, the uranium parent and the thorium-234 should have the same shape, right? The parent is producing the daughter in equal amounts, and because the daughter is decaying so rapidly that once you correct for how much uranium is there, the uranium and thorium should have the same profile shape. But what's really cool about this parent and daughter pair is that unlike the really soluble uranium, the thorium is actually very sticky. So unlike the uranium, which likes to hang out in seawater, thorium is basically hates seawater, and it will stick to anything present in seawater that's not a seawater matrix. And so this basically decaying daughter here is instead sticking to any organism or cell or small amount of organic matter that's present in the surface ocean. And as we saw before, what happens with the biological pump? What's happening to all those particles in the surface ocean all the time? They're sinking out, right? So this daughter here is basically sticking itself onto the things that are sinking out of the surface ocean. So instead of looking like this, like we might expect here, where the two things match, there's actually, due to the biological pump here, a deficit in the surface. So all these particles that are being produced at the surface are capturing thorium on them and then sinking out and bringing that thorium with them. And so the profiles of these two elements actually are separated by the intensity of the biological pump. So this deficit here is going to be much deeper and more negative if the biological pump is streaming out tons of particles, right? If the ecosystem is very not productive and only small numbers of particles are being produced and sinking out, then these profiles will be much closer towards each other. So all of a sudden we have basically an isotopic tool for looking at the intensity of a process that's very difficult to measure directly. So we basically have isotopic evidence for something that is very indirect. So this is cool. This is basically a tracer of the biological pump. And some little pictures here to show how this is done. So as I mentioned before, this daughter here, 24 days, right? Geotraces cruise, 62 days, right? So if you want to measure thorium and your first station is on day 1, by the end of the cruise, almost all of that thorium, at least only an eighth of it, is going to be left behind, right? 3 half-lives later, only an eighth of that thorium is left behind. So the groups that make these measurements actually do these measurements at sea. So not only are we at sea collecting samples for people to measure back on land, some of these groups are actually making measurements of isotopes at sea. So in the Cafe Thorium van, the group that does this calls themselves Cafe Thorium. The guy who does it and his students, he calls it Cafe Thorium because his van at sea actually has or used to have a really nice, expensive espresso machine. And so if you wanted a break, you could go into Spike's van and pour yourself a shot of espresso. It was super nice. And then someone broke it. And because Spike and everyone is near the end of the career, they're like, we're not going to spend another $2,000 on an espresso machine that someone's going to break. So Cafe Thorium doesn't have a cafe anymore. But they still make these measurements. So these counters here in the back, this is where basically the samples are put in. The thorium, those individual decays are being measured. And they do all this interesting chemistry at sea to get this signal present on the filters. So the end result of this is cool, really amazing maps of processes that are really hard to measure, things we can't measure from satellite or measure just by spending a day out on the boat. So one of the earliest, this is from 2014, only about 7 years after Geotraces started. All of a sudden, we have flux measurements. So thorium-234 is measuring the deficit in the surface here. We have measurements of how intense the biological pump is throughout the North Atlantic and South Atlantic and in really remote places of the Pacific. So we're starting to look at the intensity of the biological carbon pump. And this is really interesting because the carbon pump is what's removing carbon from the atmosphere, right? The biological pump is taking CO2 and pumping it into the deep ocean. And these numbers matter, right? So this yellow number here, around 100, is equal to about 2 apples worth of carbon per square meter of ocean per year, right? So this is it doesn't seem like much, right? It's a pretty small amount of carbon in 1 square meter of surface ocean. But you have to remember, the oceans are huge, right? So this much carbon basically spread out over a Pacific-sized amount of water is actually a huge flux of carbon. And we wouldn't have been able to make that measurement just by looking at CO2 in the atmosphere or going out on a day cruise to measure it. So these data have been super valuable. The other cool result more recently is that this is a paper from just a couple weeks ago. So this data are informing new global models of how the ocean and atmosphere and sediments all interact. So what we're seeing here, and of course the colors are backwards from what they should be, the higher flux numbers are these darker colors. But we see these really productive areas in the North Atlantic and downstream of the continents here, right? So downstream basically of Australia and the Kerguelen Islands, places where things like iron are being added in through micronutrient additions. Is basically stimulating huge amounts of productivity in the oceans. And these new models only work because we have data about how those carbon fluxes, what they actually are. Okay, so that's most of the chemistry. I want to talk a little bit about how we do it, because I know there's always interesting questions about it. So some of these technical solutions, I want to show you some of the systems that we actually use when we're going out to sea. So most of the core Geotraces measurements are made on this titanium rosette here. So it's a powder-coated titanium rosette, which doesn't contaminate for anything, no iron in it. It's been so much money on it. It's crazy. The bottles are these Teflon coated 10-liter bottles here. And you can see maybe they have these shower caps, right? So to prevent the ship's exhaust from contaminating the bottles, they actually put little shower caps on the top and the bottoms of them when they come out to deck. And then when they sample them, the people wear shower caps. There's lots of big shower cap consumption going on during these cruises. Yeah, so these bottles basically go down, fill up a sample, and come back. They get brought into a Geotraces van where a specialized team takes the samples from them and distributes them to labs around the world to make these measurements. So this is one of the systems. We also measure the nutrients. So those things that I said were measured before, we also actively measure as well because their specific numbers matter. So we have a whole separate system for doing this. This is the dirty rosette, and this is an image of its recovery here. So this is a very similar system. It's a little less clean, but we don't measure the clean things on it, so it doesn't matter too much. So this is what it's coming out. This is a pretty nice day. I would say that was a below average sea state day for Geotraces stuff. Yeah, so this system is pretty cool. It comes up into this specialized carousel here, and it has this little reticulating winch which just plucks it out of the water and puts it nicely on deck. Only one person needs to do it. The only thing this person can do wrong is not unclip it from the deck, which happened multiple times. Like go to lift it up and it's still tied onto the deck. That's one person's only job is to do that. Yeah. No injuries, no problems with that one, but yeah. Yeah, so this system is also making measurements. Then we also, like I mentioned before, all those particles and things, the iron that's precipitating out of seawater, we make measurements of those as well. So Geotraces, my group, and the groups of my PhD advisor and my collaborators use these systems called McLean pumps or water transfer systems if you're the manufacturer here. Basically a large pump that goes down and sucks a huge amount of seawater over filters. So this is how we can make measurements of things that you might need 1,000 liters of seawater to make a measurement of radium, right? So if you can measure radium on a filter, we will have a sample for you and give it to the groups that can make those types of measurements. So some of these samples get punched and go into that Cafe thorium van to be measured while we're at sea. Yeah, so the little side note here. So this is a single pump going in the water here. But they're pretty big. Spike's about 5 feet tall, but they weigh about 200 pounds. So we throw them in the water like this. This is just one of them going in the water. And this is a pretty nice day on a project called Exports, actually. And they're only putting one in. Normally we put 9 of them in the water. So I'll show you what it looks like on my normal Geotraces cruise. So this is a cast from the Southern Ocean. And Mariah, my grad student's here. This is me feeding the line. So this is a rougher day. There's snow, 30-knot winds. The sea was washing over while wearing all these like Gumby suits, trying to stay dry. So yeah, lots of different interesting sampling challenges. We also measure the aerosols. So Cliff Buck isn't here, but we use these samplers for the atmosphere, which actually get mounted on the highest point of the ship to prevent contamination. So they're measuring aerosols, and after the cruise, say in the Atlantic here, they'll figure out, oh, some of these aerosols came from North America. The aerosols from the center maybe were whipped up by the ocean. And then those dust sources that were present in Africa, those samples can be measured from aerosols samples that were taken there. So yeah, we're measuring the ocean and the atmosphere. And it's also not without its problems. So like this is a day where we didn't do any sampling on our last cruise. A pretty nasty storm in the Southern Ocean. This is the Geotraces rosette, and it has a little bandage here. And you'll note that maybe you can't see, but this pole used to be straight, and now it's bent in from a deployment where it hit the side of the boat a little bit. So yeah, not without its sampling challenges. This is the wire that broke that had my samplers on it at one point in the cruise. So this little Kevlar wire parted under tension, and we lost a bunch of McLean pumps to the bottom of the ocean. So rule that don't put something in the water that you need to get back, right? Nothing goes over the side that you might not see again. So yeah, pretty rough day on the Ravel. So yeah, so a lot of these sampling challenges are built around dealing with this types of things. So yeah, lots of fun too. So great colleagues and collaborators. Geotraces, like I said, is a huge program. This is us enjoying our first fresh vegetable after almost a month without fresh vegetables in the Southern Ocean. This is a Christmas day. I'm dressed up as Mrs. Claus. My friend here is Santa here. So yeah, a little pretty normal role. This is our chief scientist enjoying a movie. It's about 5 minutes after the starting credits started. So yeah, lots of fun. Seagoing work is great, and collecting the samples is only half the battle. We do a lot of analyses and stuff when we're back on land. The thorium group is lucky; they come back and their data is already done. But most of us actually have to go to lab for the next 2 years. So, yeah, really interesting and challenging work making these measurements. So I'll end here. I'll mention that all the data I showed today are public, available through the Geotraces intermediate data product, and there's new updates coming almost every year now. You can explore the data on your own using this program called WebODV. There's also on Geotraces website, the geotraces.org website, has descriptions of all these different tracers and what they mean and links to the studies themselves if you're interested in reading into the scientific papers. There's also some really great YouTube videos on their YouTube channel. Chemistry is cool. And yeah, I'm happy to answer any questions you might have about it. So, yeah, thanks, guys. Go for it. Oh, I saw you.
How long are your cruises?
So most of the Geotraces cruises are over a month because they're big sections. So 40 days is pretty normal. My first Geotraces cruise, the ship broke after 3 weeks. So we lost a rotor basically. So sometimes they're longer for technical reasons, but yeah, I would say on average they're about 40 days. The really long ones in the Pacific are up to the endurance. Most of the oceanographic vessels have an endurance of about 60, maybe 65 days. That's about as long as they can go before they're either running low on food or lube oil. So, yeah.
Yeah, I've got several questions.
Sure.
You mentioned and showed us examples of iron and thorium and some of the other elements, but there are lots of other rare earths and
totally, yeah.
that could be studied. Have you been studying those? Are other people studying those, or are there some that are kind of put aside and nobody's gotten to them yet?
Yeah, it's definitely a mix of both, right? So like every element in the color-coded periodic table I showed has some analyst or PI who's measuring it. Geotraces is built around these core measurements. So iron is a core measurement, aluminum, some of the trace elements are always measured on every cruise. The rare earths and some of those blue elements are only measured when there's a PI who's specifically a scientist who's specifically interested in measuring it in that ocean basin. So, but like things like particles, the aerosols, those are all measured all the time. You know what it is? When the program first started, I wrote a paper or gave a talk actually on 4 stations that were from Africa, just in that African dust plume, okay? So I had a whole scientific talk in Prague, right, about 4 elements from 4 stations, right? Now we have hundreds of cruises and sections, and there's this mad rush to keep up, right? So sometimes the sections are only a year apart, right? Sometimes some of these elements takes 2 or 3 years to measure in the lab. So we're at the point where we're actually generating data and collecting new samples at the same time. So some of these groups are totally overwhelmed. So it totally varies, yeah. Myself, yeah, the particles from those pumps, we tend to measure almost the whole periodic table. What we can do with it remains to be seen, right? So sometimes we pick the lowest hanging fruit and write paper about it. So yeah, it depends.
So another concern, when I was doing research, I was used to the concept that if I screwed something up, I could repeat the experiment the next day and do better. You're in a situation where, except for maybe the thorium, a lot of the other stuff or all of the other stuff, you've got to wait till the cruise is over, get it back on land and test it. So I'm wondering, you're using blanks to try to be sure the data is good. How often do you run into a problem where, what's the percentage of the time that the experiment is no good?
Yeah, or where a sample gets contaminated? That's a good question. Some cases, like with the water samples, most of the people who are taking water samples, they can just reanalyze, right? They'll request 100 milliliters, and their measurement might take 10. And so if they contaminate it, or the bottle, I guess if the bottle's contaminated, I guess you have to throw it away. A lot of the legwork before the first cruise even sailed was building a cookbook, an instruction manual for all these different procedures to make sure that everyone's intercalibrated and using the right procedures to collect things. And you can't get your data published in this intermediate data product without following the rules. So there's lots of different steps along the way. Just like you wouldn't just rely on one lock to keep your house locked, right? Like you'll have a door, right? You'll have a dog, right? There's lots of different steps along the way. So we're using our blanks. We're using clean procedures. Also, when the data gets submitted to the data product, a whole panel of Geotraces scientists reviews the data. The other thing about that big map too is that all these different sections, they almost always have a crossover. And so different ocean Geotraces programs, like the US and the German crews, will overlap at multiple stations. And we make sure that we get the same measurements, even if they're on different years. So yeah, intercalibration and standardization is a huge part of it, yeah.
And so in terms of data being good to publish or to put into the data bank, is it, it sounds like it's a very high percentage of the time.
Yeah, yeah.
Data.
And usually if a data point is suspect, right, like say the profile looks like this and then there's one over here and then it goes back, right? Like sometimes that sample's real, right? Sometimes it is actually like a flyer is a real data point. So the data will also get flagged. And so the data set isn't just a spreadsheet of depth and concentration. It also has information, meta information that the analyst says is like, hey, I'm suspicious of this data point. And if you want to plot the data without those points, you can look at it that way. So shades of meaning are encoded in it for sure.
And last question, you mentioned that you're also looking at organisms, and assuming some of those organisms would be phytoplankton.
Yeah, for sure.
And I'm wondering, are you or other people looking at concentrations of some of these nutrients in different conditions, different ocean conditions, different depths? Is that being actively?
Yeah, and we do it in a couple different ways, right? So like my group, we'll take that filter and digest it with strong acids and look at the whole periodic table, right? And we'll use statistical methods or knowledge of the concentrations of things or the different behaviors of the elements to figure out which is a phytoplankton signal and which is like a dirt or dust signal, right? So there's some games you can do. But then there's other groups. So the group that I did my postdoctoral work in, they actually will take individual samples of cells and analyze them for their nutrient concentrations. And so in that case, we'll take a single diatom cell and map it for its iron, its cadmium, its cobalt, and all those different elements. And so yeah, and that sometimes gives a different number, right? Like maybe you're measuring that diatom, but the ecosystem is made of a different phytoplankton, right? So it's not always easy to connect the dots, even with good knowledge of what is what. I think at the end of the day, what we're doing is, and our understanding has changed, right? Like the first cruise I went on was North Atlantic Geotraces, super dusty. Last cruise I went on was in the South Pacific, almost no dust blowing off the continents down there, right? So all these things and these interpretations that we made in the Atlantic don't necessarily apply to the Pacific. And we're learning which micronutrients behave certain ways literally just by making the measurements. So our understanding is changing as the data becomes available. So yeah, there's actually been some good debates too, right? So I didn't talk about it, but cadmium caused this like big kerfuffle where like a group basically published a whole bunch of paper with isotopes and everything, thinking that they had a whole brand new thing about cadmium. And then other groups like myself were like, I don't buy it, right? So like there were some like pretty stuffy meetings where people were talking about things like cadmium. It's like, oh, I never heard chemists get upset about this rare element on the periodic table. Eagles got pretty bruised. So yeah, interesting times. Yeah, question.
Are there corporations or entities out there essentially mining for some of the more rare elements?
Yeah, so the mining that's going on with like manganese nodules and stuff, they recruited a whole bunch or tried to recruit a bunch of Geotraces scientists to make their measurements. The European groups have strict standards for environmental impact in these new mining operations. And if you think you're polluting for manganese, like you might as well ask a Geotraces scientist to measure it for you because we're pretty good at it. Yeah, I wouldn't say, it's not like the cigarette industry, right? Like hiring a bunch of scientists to say cigarettes are cool, right? Like most of the folks, actually the woman that I know that is going is Jess Fitzsimmons, is Santa in that picture. She has done some work with the mining groups looking at contamination from those plumes. At the end of the day, it's a different set of biases in that like they're providing access to the sea and they're providing access, but a lot of scientists learned from the tobacco industry and things in the past where like they didn't sign on to make those measurements if they weren't going to be free to publish it as science. And so, yeah, it is happening, but it's a lot more believable, I think, than it would be if it was a truly corporate venture, is the way I think about it. So yeah, so there's some good coming out of it.
Have you ever, this is a different question, but have places like the South Pacific that are vastly different from like, let's say, the North Atlantic and find similar things? And I mean, why?
Yeah, for sure. So like, we'll see. The South Pacific, we have samples that are literally sitting in the lab right here, right? So one of the things I'm championing is that a lot of the same processes apply, right? So dust coming into the ocean and being sunk out relatively quickly, that should happen whether there's a lot of dust or a little bit of dust, right? So we're starting to build a rule set, in a sense, for how the ocean behaves and deals with these elements. Yeah, so sometimes we'll see basically the same profile shape, but like times 10 or times 100, right? And the interpreting it one way might work, and someone else might have data for a different element that says, oh, actually, like that's a good idea, but it doesn't explain the cadmium profile. So yeah, there's definitely a large interplay. All these cruises also have data meetings afterwards. So about 2 years after the cruise sails, we all get together to present what we have from that expedition and to sort of update the people in the room with the most knowledge, ideally before the papers even come out. So one of the big successes of Geotraces is that, I mentioned this cadmium contentious story, but most of the time we're actually consensus building and figuring out what's going on as a group and building a picture before we send these things to press. And so it's actually been really successful in that sense, right? It's engendered some challenges with National Science Foundation. These cruises are very expensive, right? And they target a very specific group of chemists who can make these measurements. So at the end of the day, we've been spending a lot of time defending that we're learning enough about the chemistry here by making these measurements. So yeah, there's definitely some politics to it too. So
One more question.
Sure.
Is there more of an effort to go to new places and geotrace that, or to go back to places?
Yeah.
There's a similar profile.
Yeah, that hits to the heart of a section-based survey like Geotraces. Really, at the end of the day, Geotraces is designed to make every one of those profiles, those sections, black or yellow, right? To fill them all in. We're not going back to the same place again and again, only if we need the sections cross over, right? So those types of studies are process studies, and a lot of the same PI, like scientists, myself included, have written proposals to do that type of study in response to Geotraces. One of the first things we learned in the North Atlantic was that we measure dust on Thursday, and it's crazy high. You measure the same element on Friday, nothing, right? So like 95% of the dust gets deposited in like 5%, maybe 2% of the days, right? So a section survey like this might capture a feature or it might not. And so one of the things we're learning is like, where do we need process studies to fill in our understanding, right? When was an October cruise the wrong date to go? And you might need to go back in March or April to make it work. Yeah, and we don't know until we do the mapping, right? It's like trying to drive a car before the tracks are down, right? So yeah, a lot of the guesses have been borne out, and a lot of the things that we thought we knew about these elements is turning out not to be true. So yeah, it's a constant back and forth about it. So yeah, that's a great question. Thanks. Cool.
Question.
Yeah.
No scientific background whatsoever.
Okay.
Okay. How does all of this impact human life and the world as we go forward?
Yeah. It's a struggle with making any kind of talk like this, right? Is how to connect it, right? Let's put it this way. If you were able to measure the atmosphere this easily, you could build models that explain how the atmosphere behaves, right? So early atmospheric scientists, or in the dawn of the satellite era, could make measurements of the atmosphere very quickly and build models, weather models, right, that affect human life, right? Weather models now, when I was a kid, like the 4-day forecast was pretty crap, right? The 7-day forecast is excellent now, right? Similarly with the oceans and the atmosphere. The oceans, albeit more slowly, constantly interact with the planet, right? So if you want to understand what is the fate of the CO2 we're putting into the atmosphere, or how will the chemistry of the oceans change if the ocean acidifies or if it warms, right? The planet is constantly we're making these changes to the planet, right? And so we're building a map of the ocean's basically chemical behaviors in order to inform those models of the future, right? That big paper that I showed that had the filled in nice no dots, right? All nice sort of modeled map. Like that's only possible understanding the oceans by having measurements of all these different things and bringing them together. So at the end of the day, I think it serves human interest because the oceans are an integral part of the way the planet behaves. And yeah, the chemistry, because it's so fundamental to ecosystem structure or ocean health, needs to be understood if we want to make predictions.
We're just trying to tie this to everyday life.
Yeah, for sure. To be honest, cadmium, you're not going to wake up tomorrow worrying about cadmium, right? But one of the things we measure was mercury, right? And we found a paper pretty early on after the first data came in from from the Pacific, that the mercury pollution that was coming out of all these different operations and productions in Asia, manufacturing processes also in South America, right, like mining process for gold, was leading to about 1/3 of the mercury that humans have released is still present in the oceans, right? We had no idea about that until we went and measured it, right? We couldn't attack that problem of anthropogenic pollutant until we knew that it was a problem, right? Like, we assumed this mercury was going into the oceans and was gonna be gone forever, right? But its chemistry showed that it's hanging around. Same thing with the lead, right? Like, if lead is affecting fish or changing the structure of the ecosystem in a way, or any of these elements, we won't know until we measure it, right? So yeah, I think, yeah, it's a bit of a chicken and egg, but yeah, I agree. I mean, it's a couple steps away, but I think it's still important. Yeah.
Do you have a group that helps you when you're doing your research after you come back?
Yeah, I mean, the other scientists, yeah. So my lab group is just Mariah and me right now, right? So Mariah is my grad student sitting right behind you. But yeah, like all my colleagues and peers, everyone who sailed is interested in making good science out of it. So yeah, I mean, the Geotraces community in general is super supportive, right? I mean, like, not only in the sense that we have our own like little town halls at big meetings and stuff like that, but yeah, I mean, like, we champion our own papers, and we're, you know, if I have a question about the iron data that someone collected from India, I can send an email and they will get back to me about it. You know, like, ocean is a huge place, but the oceanography community is actually pretty small. And so, yeah, that's one of my big come-aways from this, is that, like, oh, actually, yeah, respect my peers in that sense and know that they're available, yeah.
I also have no scientific background, but my sense of the ocean is that it's always moving. The circulation and how rapidly all the currents are moving around the Earth. How does that affect your measurements?
You know, so it depends, right? So some of these elements, like the macronutrients, the ones that have thousands of year residence times, they're super affected by circulation, right? Some of the particles, the things that are sinking out, they're not affected by circulation at all, right? That particle that was produced at the surface, when that organism dies, it sinks to the bottom in a day or two, right? So circulation might move it maybe a couple hundred meters from where it was formed. So it really depends, right? The interplay between the physics and the chemistry and the biology is totally element specific, and also something that we, you know, that's part of the magic of the program, is to sort of figure out to what extent it does. Neodymium is one I didn't talk about either. Neodymium is this element that has different concentrations in different continents, right? So Antarctic neodymium and you know, Pacific Rim neodymium is much different from old continents like Canada and Northern Europe. And so neodymium is like super affected by circulation. And one of the early, neodymium is a core measurement in Geotraces. One of the first things they did when they had data from every ocean basin was reconstruct the circulation of the oceans in the past based on the data we had from the modern era for neodymium, right? So not only are we learning about the way the oceans behave, but because some of them are affected by circulation in the past, we can be using them to answer questions about past circulation too. So yeah, it really depends, so yeah. Some of I like to focus on the ones that are like a little easy. Circulation messes things up, so I mostly focus on the particles. The particles don't, yeah, the less physics, the better for me, if you ask me, but. Yeah, great questions. So I put my email up. If you want to follow up, by all means, do so. But I think we're getting pretty close to conch out time for most.
We don't have any questions online, so
Okay, great.
you're okay there.
Awesome. Well, thanks, guys.
Thank you.
Yeah, and I think we have our next month, right?
Next month is a open lab night when we will have a subset of faculty in their labs to talk to you about their research and kind of show their labs to you. So we'll be announcing what labs those are, but it'll be on the second Tuesday at the same time as usual.
Cool.
So we encourage you to come, and we look forward to seeing you again.
Yeah.
Great.
Cool. Thanks, guys.
Thank you.
Thank you very much. Would it be possible